Hi, Professor.
Hi, Hayden. Hope you're doing well. We'll get started in about 3 minutes.
After our lecture today, I did have a few questions on last week's lecture. When you had that picture of the two genes, I had like a bunch of questions on that after.
Okay. I'll be sticking around after class, after a small break, and we can go over some things.
Okay.
Okay, good afternoon, everyone. Welcome back. I hope you're doing well. I hope you had a nice weekend. We can see from the news media that things are getting increasingly worse in Pennsylvania, so please do everything you can to keep yourself and your family members safe at this time. Okay, from last week, from the last email I sent out, I did send out a study guide for your second lecture exam, which is coming up on April 9th. If you did receive the study guide, please type yes into the chat window. Okay. All right, so Kevin and Sean, okay, Jose, very good. All right, so I know that you folks did get that. Do you have any questions about the study guide? I tried to make it as self-explanatory as possible, but do you have any questions about the study guide before we dive into more content here today?
I didn't get it.
Okay. Hayden, are you getting emails from me that I'm sending to your hack account?
Yes.
Okay, so check last Thursday's email, which would have been the 26th, March 26th. If you didn't get an email from me, then check your spam folder. And if you still didn't get it, then send me an email and I'll resend it to you.
Okay. Yeah, I don't have any emails from you from the 26th, but I'll check my spam.
Okay. Okay. Other questions about any of the content from the study guide? Okay. Well, as you folks really dig into study and bear down for it, if you do have questions, feel free to send them to me and I'll try to address them for you. Okay. So any questions about the class or the semester, anything going on that you want to ask right now before we start new content?
So I do have a question regarding the first literature review. I was just curious when we might expect to see a grade for that.
Yes, Kevin. They are all graded. I just have no way of physically getting the documents to you to see my comments. I suppose the best thing that I could do right now is take your hack numbers, your H numbers, and put them next to your grades in an Excel document and send it out to the class. That way, you would be able to identify your score based on your H number. Would anybody have an objection to me doing that? Okay. All right. So that's what I'll do, Kevin, is I will, in the next day or two, I'll put together a small Excel spreadsheet with your literature review grades in, and the grade will be next to your H number from the hack ID. Okay. I wish there was a way that I could physically get the papers to you, but I don't think there is at this time. Okay, any other questions? Okay, gang. Well, let's get back into it. I'm going to spend maybe 20, 30 minutes here on chapter 9, close up shop on that and get into chapter 10, which is translation. So let's begin. I'm going to start sharing my screen with you again, as I usually do, so you can see the Microsoft Word document. Okay, so on my screen at this time, you should see the last notes that I believe we took from last time talking about the process of transcription. It was number 4, and it was our description of elongation. And the last note was that E. coli RNA polymerase can manufacture RNA at a rate of 40 to 50 nucleotides per second. So if you can't see something or I'm going too fast, please type into the chat window or unmute your microphone and just speak up. That's quite okay. All right, so we're continuing our conversation about transcription here. And I will start with number 5 here, which should be some new notes for you. Okay, so we've just explained elongation, okay? Number 5 is that RNA polymerase continues making this new RNA until it reaches a termination signal within the DNA template. Okay, so RNA-P continues making new RNA until it reaches a termination signal, which destabilizes the enzyme. When I say destabilizes the enzyme, I mean it destabilizes RNA polymerase, which destabilizes the enzyme, causing it to pause and fall off the DNA template. Okay. Now, a couple subpoints here I'd like to make for you. That is that A, unlike the promoter, which is not transcribed into RNA, the termination signal is transcribed into RNA. Okay, so that's an important distinction to be aware of in your mind, that the termination signal actually is transcribed into RNA, unlike the promoter sequence. Secondly, as the terminator RNA is synthesized, okay, its unique sequence, okay, so the terminator sequence is going to be a specific word, if you will. It's a DNA word that is then transcribed into an RNA word. Its unique sequence allows the terminator to fold back on itself, forming what's called a hairpin structure. As the terminator RNA is synthesized, its unique sequence allows the terminator to fold back onto itself, forming a hairpin structure. Okay.
When you say DNA word, I thought that only RNA has codons to describe it.
Okay. Thanks for asking, Hayden. Yes. No. I think you may be confusing some ideas at this point. Codons are found in the RNA, and they're used by the ribosome to translate and manufacture proteins. But when I talk about a DNA word, I'm talking about something like the origin of replication, which we used when we were talking about the DNA manufacturing process. And a promoter in the DNA, which is the landing target for RNA polymerase, is also a DNA word. So a DNA word is simply a stretch of DNA nucleotides that acts as a binding site typically for a protein. If we have an RNA word, it's a stretch of RNA nucleotides, which is a binding target for a protein that attaches to the RNA. So we can have DNA words, we can have RNA words, okay? There's nothing wrong with that terminology. But when you start talking about codons, you're talking specifically about the genetic code, which is defined as part of the RNA that a ribosome will decode to make protein.
Okay.
Okay. Okay. Anything else right now?
Your screen's a little blurry on the Word document. I can't really see it.
You can't see it. Are the rest of you having a hard time seeing my Word document? Okay. Would it help? Okay. Would it help if I made it a little bit bigger?
Maybe.
Okay. I noticed also when you logged in, Hayden, that you were sitting in front of a window where there's a lot of light coming in your window. Could that be causing a problem for you?
No, it's the screen. The words are just really blurry. It wasn't so blurry before, but now it's really blurry. I'm not sure.
Okay, so let me minimize the window and open it back up to see if that makes any difference.
Okay.
So it's crystal clear on my screen, and unfortunately, it looks like most of the other folks are seeing it clearly. Are you going to be able to follow along?
Probably not.
Okay.
But if it's recorded, I could always just go back.
Okay. It is recorded. If you wanted to log off and try logging back on, that would be okay, because we'll be right here, okay?
Okay. Yeah, I'll do that. And I'll turn off my Wi-Fi and turn it back on.
Okay.
Might be that.
All right, folks. So continuing with bullet point 5B. As the terminator RNA is synthesized, its unique sequence allows the terminator to fold back on itself, forming a hairpin structure. And this destabilizes the RNA polymerase, okay, which destabilizes RNA polymerase, okay? So in some genes, the hairpin structure is enough to terminate, meaning finish, meaning end, meaning stop, the transcription process. Okay? In other genes, the hairpin structure is used by another protein called Rho to actually disrupt RNA polymerase, and stop transcription. I believe that was hinted at in one of the videos that I showed you last time, and certainly some of the videos I sent you. I think one of them did mention the Rho protein, okay? So there are some genes that will terminate without, there's some genes that the transcription will terminate without Rho, and in that case we have, let me tack this on here, we have Rho independent termination. And in the genes where we do need Rho, we have Rho dependent termination. Not a big deal right now. It's certainly not, something you need to worry about too much, but I'm just making you aware of it. Okay. All right, gang. So that basically covers prokaryotic transcription at this time. And what I'd like to do next is talk about some important differences with eukaryotic transcription. But before I do that, let me ask you, do you have any questions about the prokaryotic transcription? Any questions about prokaryotic transcription coming out through the chat window? So obviously during your personal study time, you're going to want to sit together with the animations and the notes and step through it step by step. Make sure you try to understand the major themes and points of each major process, initiation, elongation, termination, that kind of thing. All right, not seeing any questions. I'm going to continue, gang, so we can move on here. All right, so I'm going to give you a few notes about eukaryotic transcription differences between eukaryotes and prokaryotes. Eukaryotic transcription differences. Okay, number one. All right. Eukaryotic cells have 3 different forms of RNA polymerase. Bacteria, most prokaryotes that we have identified, only have 1 version of it. Okay. Eukaryotic cells have 3 different forms of RNAP. Bacteria typically have only 1. Okay. And Here, I'm going to put in your notes. One of the tables. Hold on a second here if I can find it. Yes, okay. If you have your textbook available, table 7 of page 252 is what I would want to highlight for you. Okay, and so let's do it this way. Okay, table 7 of page 252, and we'll mention that RNA polymerase 1 manufactures most of the ribosomal RNA inside a cell. Okay, RNA polymerase 1 manufactures most of the ribosomal RNA, and since ribosomal RNA is made in the nucleolus, this is where you can expect RNA polymerase 1 to do its work. Remember, ribosomal RNA is made inside the nucleolus. Second thing, RNA polymerase 2. RNA polymerase 2 is responsible for making the messenger RNA and small, tiny little fragments of RNA that are called small nuclear RNAs. Manufactures most messenger RNA and SN RNA. SN is short for small nuclear RNA. Okay. And so this is, of course, going to take place in the nucleus, but outside the nucleolus. And then RNA polymerase 3 is responsible for making the 5S subunit of ribosomal RNA as well as transfer RNA. Okay, and that will also take place in the nucleus, but outside the nucleolus. All right, so pop any questions up if you may have them, gang. All right, a second point that I would want you to appreciate. Transcription in eukaryotes requires that the chromosome material be in its uncoiled chromatin state. Okay, that's even more important in eukaryotic cells than it is in prokaryotic cells. Transcription is heavily regulated by the structure of the chromosome versus chromatin. When DNA is loose, as in chromatin, it is much easier to perform transcription. Number 3. Eukaryotic genes typically have more complex regulatory information than prokaryotic genes. And this is a theme that's going to continue into the course as we get deeper into the course and start talking about some eukaryotic gene regulation, but we're a few chapters away from that. Eukaryotic genes often have more complex regulatory information, things that are present that you may have heard about from other courses or other biomaterial you've read on your own, things called enhancers and silencers. Eukaryotic genes often have more complex regulatory information, such as enhancers and silencers, which are sequences. They are effectively DNA words. Enhancers and silencers are DNA words. They are nucleotide sequences that additional regulatory proteins combine to and further modulate the transcription process. Okay, so enhancers and silencers, which are sequences that additional regulatory proteins bind to modulate the transcription process. Number 4, things to keep in mind here during your personal study time. Eukaryotic transcripts, okay, particularly, well, let me ask you a question, folks. Question coming through the chat window, okay? First question, what process makes messenger RNA? Go ahead and answer that. What process makes messenger RNA? I'm not trying to trick you, I promise. What process makes messenger RNA? Okay, thanks, Jose. It's in fact transcription, right? It's the topic of our conversation right now. Let me ask this. What process makes tRNA? What process makes tRNA? Still transcription. Very good, John. Let me ask you this. What process makes ribosomal RNA? What process makes ribosomal RNA? John says it's transcription. He's right. Let me ask this. What process makes peewee RNA? Peewee RNA is a real thing. What process makes peewee RNA? Transcription. Okay, gang. So what point am I trying to make to you? That if we're manufacturing RNA, no matter what kind of RNA it is, transcription is the process used to manufacture it. Okay? So when I say eukaryotic transcripts, for my next point here, bullet point number 4, I'm going to have to specify something. Eukaryotic messenger RNA transcripts are stabilized by the addition of eukaryotic messenger RNA transcripts are stabilized by the addition of a 5' cap, okay, which is made of 7-methyl-guanosine, often abbreviated 7 lowercase m, capital case G, 7-methylguanosine. So the 5' cap helps to stabilize it. The presence of that 5' cap is going to ensure that the messenger RNA half-life is increased, okay? And secondly, a 3' poly A tail, okay? Both A and B here, both A and B increase the half-life of a population of messenger RNA molecules, okay? Right? So let's say that we have a population of 1,000 molecules whose half-life is 30 minutes. That means after 30 minutes, I'm only going to have 50% of the 1,000 or 500 molecules left. But if I were to add a 5' cap and a 3' poly A tail there, I might very well increase the half-life of that population. So after 30 minutes, rather than having 500 messenger RNA molecules left, I might have 750 messenger RNA molecules left. Okay? Gang, are you okay?
I'm okay with that, but I was wondering if this can be like used as part of regulation. Like, do we sometimes not put those caps on in order to decrease the half-life?
John, I can barely hear you. Please type your question into the chat window. I think it had something to do with regulation. Okay, so John's asking, is this part of regulation? Like we cannot include these caps to decrease the half-life? Yes. Okay, the basic answer to your question is yes, that the presence of the cap, whether it's present or not present, can be a regulatory strategy. And actually the length of the poly A tail, you know, whether I have a 20 nucleotide poly A tail or a 50 nucleotide poly A tail makes a big difference. So yes, this is part of regulation. Other questions? Okay, gang, let's keep going here. Let's see, I'm going to go to bullet point number 5. For bullet point number 5, okay, I want you to appreciate that eukaryotic promoters have a consensus sequence that is called a TATA box. Okay, we mentioned the TAAT sequence of a prokaryotic promoter and how that is not technically a TATA box. Okay, TATA box is a phrase used for eukaryotic promoters. Eukaryotic promoters have a consensus sequence called a TATA box. Okay. This is not to be confused with the same TA sequence of prokaryotic promoters, which you recall is also called a Pribnow box, not to be confused with a Pribnow box from prokaryotic promoters. Okay. The TATA box of the eukaryotic promoter is also known as a Goldberg-Hodgeness box.
Excuse me.
Yes, Hayden.
Going back to bullet point 4, when you're talking about adding the cap and the polytail, are you talking about pre-messenger RNA?
Well, you're definitely in the ballpark there. Once the messenger RNA is mature, so remember that in eukaryotic systems, the introns are going to have to be removed and the exons spliced together. Once that has occurred and you have the mature RNA, the mature messenger RNA, then you're going to add the 5' cap and the 3' poly A tail to the mature messenger RNA.
Oh, okay.
Okay, so the TATA box of a eukaryotic promoter is also called the Goldberg-Hodgeness box. Okay, this DNA word is located about 30 nucleotide pairs upstream of the transcription start site. Okay, so Hayden's question was actually very good timing, because I also intend to put into your notes the fact that you do definitely want to realize that eukaryotic transcripts typically contain introns, which are not very commonly found in prokaryotic genes. It's not that we never find them in a prokaryotic gene, it's that they're pretty rare compared to eukaryotic cells. Eukaryotic transcripts usually contain introns that must be spliced out to form mature messenger RNA. And remember, prokaryotic cells prokaryotes typically do not use introns. Okay. Last thing here, we made a big to-do of it at the beginning of our note-taking for this chapter, or at least I think we did, that in prokaryotic cells, transcription and translation are coupled because prokaryotes don't have a nucleus. And that means in eukaryotic cells, transcription and translation is uncoupled. Okay. But uncoupled, not incoupled. But remember, in eukaryotic cells, transcription and RNA processing, the so-called splicing that's in the textbook that you're reading, the transcription and RNA processing / RNA splicing is coupled. Okay. All right, gang. Let's see here. Now, you can see how beefy these topics are. I could spend the rest of the class period talking about splicing, and I don't want to do that. Okay, we have other things we have to talk about. You should be familiar enough with RNA processing to know a basic definition of what it is and to appreciate that eukaryotic cells have to perform RNA processing in order to generate mature RNA. But I'm not going to beat you up on your upcoming exam with it. And as you've already seen from the study guide, I do not have an essay on RNA processing. Okay, there's plenty of material that I can challenge you with for DNA replication, transcription, and translation. I don't need to add in RNA processing or recombination, which would just make things unreasonable at this time. Okay, so what I will do is in the next email I send you, I will send you a couple videos on RNA processing and RNA splicing that I think are particularly good videos that if you're intellectually curious about these processes, if you want to understand them maybe a little bit better than your book is describing them, I'll recommend that you take a look at these things. But the splicing mechanisms that we would need to talk about are every bit as complicated as our conversation was with recombination. And at least right now, I just don't have the time to talk about it. So, okay, gang, that's basically where I'm going to let chapter 9 at this time. Do you have any questions you'd like to ask before we start delving into chapter 10? Okay, not hearing anything, not seeing anything in the chat window, so I will assume that you're okay. I hope you're okay. And as always, if things come up during your personal study time, take note of it so that you can send an email or ask it at some point. All right, gang. So that's our coverage of chapter 9. Let's start going into chapter 10. Chapter 10 is the last chapter. Chapter 10 is the last chapter that you are responsible for the upcoming lecture exam, so we definitely need to get through it. All right. Chapter 10 is, of course, about translation. Bear with me just a moment while I get my materials organized here. So chapter 10 in your textbook starts on page 269. And as I usually do, I want to start our conversation on chapter 10 with some foundational information that you always want to keep in the back of your mind while you're reading and studying. Okay. Well, let's state the obvious. The process of manufacturing protein is called translation. You know, if you look at anatomy and physiology textbooks and things that try to describe this in a basic way to students, they always say that the process of manufacturing is called protein synthesis. Well, that just makes my skin crawl because, come on, it's translation. Okay. Okay. Now, I'd like to point something out. If you do have your textbook available, the current edition of the book, the opening page to this chapter, where the chapter is titled Translation in Proteins, and if you have the current version of the book that I recommended on page 269, your textbook makes this statement. And I'm just going to read a sentence or two from your own textbook. So I'm on page 269, which is the opening page of chapter 10, Translation in Proteins. Okay. One of the first sentences on that page is this. It says, however, the final product of gene expression in most instances is a polypeptide chain consisting of a linear series of amino acids whose sequence has been prescribed by the genetic code. Folks, that is wrong. That is no longer considered correct. Okay? So I want to take note of something that is no longer considered correct in your textbook. Okay, so a historic errata from the textbook, and if you have the same version that I do, I'm talking page 269. Okay, and I want to type that in so you know what I'm talking about. However, the final product of gene expression in most instances is a polypeptide chain consisting of a linear series of amino acids whose frequencies, whose sequence has been prescribed by the genetic code. Okay, this is no longer valid. This is no longer valid. Okay, we now know due to research, within the past 15 to 20 years, that in fact most of the DNA inside of a cell, most of what we would call the genome, is in fact transcribed into RNA of one form or another, but very little of it is actually made into protein. Okay. Modern experiments reveal that most DNA inside a cell is made into RNA, but very little RNA is made into protein. So what we used to think is true is just not true anymore. Okay. And so let me just, I'm going to be coming back to the Word document, but I'm going to leave it for just a moment to show you an example of the research that kind of proves my point. So I'm going to go to, NCBI PubMed and look for an article here. This article is called Genome-Wide Transcription and the Implications for Genomic Organization. Oh, good, it's there. Okay. So, all right. So this article I usually bring up when I'm face to face with you. Let me just read the abstract for us. It says, recent evidence of genome-wide transcription in several species indicates that the amount of transcription that occurs cannot be entirely accounted for by current sets of genome-wide annotations. In other words, the RNA molecules that we have annotated and described in our computer databases are far fewer in number than the actual number of RNA molecules that can be detected as being made in real cells. Continuing with the abstract, evidence indicates that most of both strands of the human genome might be transcribed, implying extensive overlap of transcriptional units and regulatory elements. These observations suggest that genomic architecture is not collinear, unlike what your book kind of suggests is true, but this is how it goes, but is instead interleaved and modular, and that the same genomic sequences are multifunctional. That is, used for multiple independently regulated transcripts and as regulatory regions. Question: what are the implications and consequences of such an interleaved genomic architecture in terms of increased information content, transcriptional complexity, evolution, and disease states? That's an open-ended question. Nobody knows the answer to that. It's a topic for continued research. But gang, I simply wanted to point out that it's not just your teacher telling you that something's wrong in the book. There's actually scientific literature to support that maybe we need to think differently about this than the current textbook is telling you. Okay, so thoughts or questions? Don't worry about that article. You don't need to read it or anything. I mean, if you want to, you could, but you don't have to. So the take-home message, gang, is that most DNA inside a cell is in fact transcribed, but very little of what is transcribed gets made into protein. So the big 800-pound gorilla in the room for researchers is, what the hell is all this RNA? What's it doing? What is its job in the cell? And, you know, while we don't truly know the answer on that, certainly part of the answer is a lot of the RNA that does not code for protein actually is important to regulate other processes inside the cell. Okay. I'll put that as a second bullet point. At least some of the RNA that is made but not translated is used by cells to regulate processes other than translation. Okay, gang. Thoughts, questions, confusion? Okay, Hayden, I'm typing too fast. Thank you. Let me know when it's okay for me to continue.
I do have a question, Dr. Allen. So what would be an example of some of these processes that would be regulated by RNA other than translation?
Okay. Chromatin and chromosome structure is often modified by proteins that bind to the DNA. But the question becomes, how do the proteins know where to bind? Typically, they bind because there are small pieces of RNA called guide RNAs that act, as the word suggests, they act as a tour guide for the protein to bind to the correct target on the DNA to modify the actual structure of the DNA. Anything else? Is it okay for me to keep typing, gang? So John has a question. The RNA that's used for regulation is what explains the gap between the amount of transcription and translation. I would say, as far as we know right now, that yes is probably the correct answer to your question, John. Hayden, are you okay for me to continue? Okay. I'll try to slow it down just a little bit here. Okay, so bullet point number 3. So I wanted you to know this. You need to, at least I try to think it's important for me to point out some times when maybe the book isn't as current as it needs to be. But let's face it, all textbooks are somewhat dated by the time they get published. That's just how it works. It's really a hard job to make a textbook and keep it as current as possible. All right, so we are in chapter 10 talking about translation. The next foundational bullet point I would want to do is simply define the 3 major types of RNA that are important to the translation process. Essential to translation is 3 types of RNA, and you know what they are. I mean, if you've had Bio 101, you know what they are. It's the messenger RNA. All right. This is the molecule that's made from the template strand of the DNA, which is the gene itself, which contains the information needed to make a specific protein. Messenger RNA is the molecule transcribed from a gene which contains the information needed to make a specific protein. The second major form of RNA is the transfer RNA, the tRNA, okay, which you know is also made by the transcription process. This is the molecule which delivers amino acids to the A site of a ribosome. Okay, the A site has different words, has different terms associated with the letter A, but for right now, if you want to think of A as the acceptor site, that would be okay. Okay, and so let's see here. Yes. Okay. All right. Continuing forward here. Letter C is the ribosomal RNA, the rRNA for short. This is the molecule which forms the enzymatic guts of the ribosome, and which is actually responsible for linking amino acids together with peptide bonds. Okay, so ribosomal RNA is the molecule that as you'll learn in just a moment, there's actually different forms of ribosomal RNA in the ribosome structure itself. And one of those specific ribosomal RNA molecules is responsible for the peptide bond formation. Okay. I'm just going to take a little pause here, so I know that you folks have to write these things down, and that's good. It's important that you do the writing. It will help keep it in the head. Okay. Bullet point number 4. You need to appreciate that there certainly are similarities, but there's also some important distinctions between prokaryotic and eukaryotic ribosomes. Figure 1 on page 270 of your textbook has a nice comparison of the prokaryotic and eukaryotic ribosomes. I want to give you just a couple little notes about each of those. Prokaryotic ribosomes are made of, they are made of 52 ribosomal proteins. Okay. My personal PhD dissertation was on a ribosomal protein called L4, which is the third gene in an operon of genes called the S10 operon that in bacteria is able to regulate the production of other ribosomal proteins. So long story short, you know, this is a big deal to scientists. Okay, so 52 ribosomal proteins make up the prokaryotic ribosome. That number 52 is not set in stone. Some people might say 54, some people might say 50, some people might say 56. The number of proteins that make up the ribosome is difficult to definitively define because when you purify ribosomes biochemically, the proteins that show up with the ribosome can be different from one purification process to the next. So roughly 52 ribosomal proteins in a prokaryotic ribosome. There are 3 different RNA molecules in the ribosome, and they are the 23S ribosomal RNA. The 23S ribosomal RNA is the RNA molecule that's responsible specifically for peptide bond formation. Creates peptide bonds between amino acids. Next, we have the 16S ribosomal RNA. Its job is to bind to an RNA word. Its job is to bind to an RNA word in the messenger RNA called the Shine-Dalgarno. Okay, so it binds to the Shine-Dalgarno sequence, and purpose of the Shine-Dalgarno sequence is to provide a binding target for the ribosome so that it can position itself properly to begin translation. Okay, binds to an RNA word called the Shine-Dalgarno sequence, which helps position the ribosome to translate properly, and it helps the 16S ribosomal RNA also helps to stabilize the codon-anticodon interaction at the A site of the ribosome. Okay, so I'm going to just pause because I know I was buzzing along there pretty good. The 16S ribosomal RNA binds to an RNA word called the Shine-Dalgarno, which helps position the ribosome to translate properly and helps stabilize the codon-anticodon interaction at the A site of the ribosome. All right. The third final RNA molecule that's part of the prokaryotic ribosome is the 5S ribosomal RNA. And believe it or not, after probably 50 or more years of research, the exact function of that piece of RNA is still a little bit unclear. We don't know exactly what its purpose is, which suggests to us that it may have a function in the overall stability of the ribosome, or it might be an evolutionary fossil that's really not, it's not really needed anymore. We're just not sure. Precise function is unclear. Okay. All right, gang. Let's go and just make a few, let's just make a few statements about the eukaryotic ribosome so you have it in your notes. So number five, the eukaryotic ribosome. Before we go any further, do you folks know what the S in 16S refers to? Coming through the chat window, do you know what S in 16S refers to?
That's a unit of measurement, the Svedberg units or something.
Yes, good job, Kevin. That is a Svedberg unit. It's a unit of sedimentation during purification studies. When you try to physically purify molecules, you'll get a sedimentation value. The S is the sedimentation value. It is a Svedberg unit. Okay. So the eukaryotic ribosome is made of, it's made of 79 ribosomal proteins, and it is made of 4 different RNA molecules, of which I won't define them all for you like I did the prokaryote, but I can tell you what their sedimentation sizes are. They are 28S, 5S, 5.8S, and 18S. Okay. Gang, any questions right now before we continue? So let me just put my comment about S in the chat window. S is short for Svedberg unit, which is a sedimentation. Okay. Gang, do you have any questions right now before I continue?
Does that say 28S for the first RNA molecule?
It does. It's
Okay.
28, followed by 5, followed by 5.8, followed by 18. Okay, gang. Let's continue here. Number 6. Okay, all ribosomes, as you probably recall from your Bio 101 and 102 days, are made of the molecules that we just listed, but these molecules themselves are assembled into 2 subunits, a small subunit and a large subunit, okay, which you can see highlighted on figure 1 of page 270. Okay, so the small subunit is made of ribosomal protein and ribosomal RNA. The large subunit is made of different ribosomal proteins and different ribosomal RNA. Exactly what that composition is, we'll leave for your graduate work in the future. But I would like you to appreciate that the small subunit is responsible for decoding or interpreting the codons in the messenger RNA. Okay. The small subunit is responsible for decoding the codons in the messenger RNA. The large subunit is responsible for the actual peptide bond synthesis. Okay. All right. Third bullet point, 6C. The complete ribosome, where we have both subunits together. Maybe not today, I'm not sure, but certainly next time I'll be showing you a video or two about translation. You know, scientists have been working on the actual three-dimensional structure of ribosomes for a long time. It's a hot topic of research. And one of the most pragmatic reasons for that is because we know that most of our classical antibiotics, that science has discovered over the years actually works by disrupting the structure of a ribosome to successfully and precisely kill bacteria, which are prokaryotic cells, while leaving human cells intact. So ribosome structure is a big deal. Scientific community has put tremendous person power and people years into doing this research. So the complete ribosome, where we have both subunits together, is made of 3 rooms. Now, not literally rooms, but what I envision, let me finish my notes, then I'll talk. I'm made of 3 rooms called the E, P, and A sites. Okay. Imagine for a moment that we're back on campus at our building in Lancaster, at the Hack campus in Lancaster, where we normally meet together. Imagine that building is in fact an entire ribosome, and every room in that ribosome is a pocket of space, a cavity, a canyon, a crevice, if you will, for unique activity. For example, continuing with the analogy, we would have lecture in room 229. Well, room 229 might be analogous to the A site of the ribosome. Okay, room 229 is part of the physical building. The A site is part of the physical ribosome. And then we would have our lab in room 218. So room 218 might be the P site of the ribosome. So think of a ribosome as this entity that actually has pockets of space in that different biochemical processes actually happen in. Okay, so the E site is short for the exit site. Okay, this is where the uncharged tRNA leaves the ribosome. The P site, okay, if you're completely flummoxed about what I'm talking about here, let me hint at some of the stuff that we're heading towards on page 274 and 275, where you have to appreciate the structure of the ribosome so that you can see, in particularly the E, P, and A site. On either page 274, 275, you definitely see the P and A sites that are drawn in the cartoons of figure 7. The P site is short for the peptide site. This is the location where the growing peptide chain is held in position. And then we have the A site. I need to make my notes better here for you. The A site, this is the acceptor site, for lack of, there are some more biochemical definitions for this, but I think A site suits us well right now. This is the location where the charged tRNA binds. Okay. So I'm going to put a question in the Word document for you. What is the difference between a charged and uncharged tRNA? Go ahead and answer that in the chat window if you can. What's the difference between a charged and uncharged tRNA? John LaRusso says, wouldn't charge have one less electron? Okay, that's a reasonable statement with the education that you have, John. It's not a bad guess, but that's not what it means. Okay, Sean, thank you very much. Uncharged tRNA doesn't have an amino acid attached. Exactly, okay? So charged and uncharged here is not really referring to the electrical status of the molecule. It's referring to whether there's an amino acid present on the tRNA or not. Okay, charged tRNAs are carrying amino acids. Uncharged tRNAs do not have amino acids. Okay. All right, gang. What I want to do next is go over the chemistry of peptide bond formation with you. And so this is one of those things that I'm going to have to do a horrendous drawing with you for. Okay, so bear with me. I'm going to switch gears to a PowerPoint window so that we can do a little bit of drawing. Okay, so let's put in the Word document peptide bond C drawing. Okay, peptide bond C drawing. And here we are. Okay. Let's be sure that we appreciate what a peptide bond really is. All right. So, okay. So, okay. The chemical nature of peptide bond formation. Let's see if we can get this finished today so we can go on to deeper things next time. Okay, so please bear with me here. This is going to be a little rough. Okay, so if we have two amino acids, the general structure for amino acid looks like this. We have a nitrogen atom linked to a carbon, linked to another carbon. The last carbon has a double bond to oxygen. The middle carbon is bonded to hydrogen. The nitrogen on the end is linked to two hydrogens. The middle carbon is linked to what's called an R group. An R group would be a collection of atoms, and I'll come back to that in just a moment. The last carbon is also connected to a hydroxyl group, OH. Okay, so that's the first amino acid. And if we're going to link that to another amino acid, well, I need to draw another amino acid in here. So we have another nitrogen, link to carbon, double bond oxygen. Middle carbon's got a hydrogen. Nitrogen has 2 hydrogens. Middle carbon has another R group. And the last carbon has its hydroxyl group. Okay? So the R group conveys the identity of the amino acid. Okay? So over here where I'm circling R, and over here where I'm circling R, these R groups and the groups of atoms that make up each R group define the identity of each amino acid. The atoms of each R group define the identity of each amino acid. For example, if R was a single hydrogen atom, if R was the atom hydrogen, then the amino acid would be glycine. Okay? Alternatively, if R were CH, which itself was attached to 2 methyl groups, CH3, CH3, if it was that group of atoms, then the amino acid would be valine. Okay? Please unmute your microphones and speak up if I'm confusing you. Okay, so what we're talking about is the peptide bond formation. So we're going to link these 2 amino acids that we've drawn here. We're going to link them together. Okay, and so I'm going to move out this way so I have a little bit more space. Okay, but we're going to have peptide bond formation here. Please bear with me maybe for another minute or two. So peptide bond formation, which is catalyzed, as you know from your notes, by 23S ribosomal RNA in prokaryotes, or 28S ribosomal RNA in eukaryotes. Okay, so what we're going to have here is a dehydration synthesis reaction, which I'm sure you've all heard of before. Dehydration synthesis, where we collectively remove atoms from the 2 amino acids that can join up to form water. Okay, so we're going to end up producing water in this process. And so let me change my ink color here. Okay.
What's the name of the other amino acid?
Well, okay. In our example here, let's say that the left amino acid is glycine. That's the first one. The second, more complicated one with the bigger R group is valine, V-A-L-I-N-E. Okay, so for the dehydration synthesis, oh dear, having a tech problem. Sorry, gang, one moment. Okay, So in other words, try to wrap this up. Here's an OH group and the H group. These are the atoms that are going to get pulled off each amino acid to form the water during the dehydration synthesis. And what we're going to end up with is chemistry that links each of these amino acids together. So let me draw it down here. We're going to end up with nitrogen, carbon, carbon, and now that carbon is going to be linked to the following nitrogen, then carbon, then carbon. All right, let's finish this out. Just a minute or two. Double bond oxygen. OH down here, nothing unusual. Hydrogen over here. R group there. That first nitrogen where you see my cursor is going to have an individual hydrogen atom. The carbon to the left of it is going to have a double bond oxygen. Carbon to the left of that will have a hydrogen. The nitrogen on the left end will have its two hydrogens, nothing unusual there. And the carbon where my cursor is will have its own R group. And the point here is that this thing right here is the peptide bond. This that I'm highlighting in green is the peptide bond. Linking the two amino acids that we started with. And it also shows you things that you already know from other courses, that this this linked molecule at this point has its amino end. The NH2 here is the amino end of the peptide. And over here we have the carboxy end, what's called the carboxy end of the peptide. It's very important that you appreciate, and we'll finish our conversation today with this last thing. It's important that you appreciate that when a ribosome attaches to the messenger RNA, it always attaches to the 5' end of the messenger RNA, and it synthesizes the new polypeptide in an amino to carboxy direction. Okay, so let's write that down. When ribosomes translate, they attach to the 5' end of the messenger RNA and physically move from amino to carboxy. Okay, so remember from our conversations with DNA replication and transcription, I kept saying, and I'll say it again here, that DNA and RNA polymerases synthesize new RNA 5' to 3'. In an analogous fashion, when ribosomes translate, they make new polypeptide, they make new protein in an amino to carboxy direction. Okay, gang, thanks for bearing with me on that. Gang, we're 5 minutes over our time, so I want to honor that and not hold you too much longer. Let me just make a couple points. We're going to really dig down into the translation process next lecture period. I'm a little bit behind schedule on grading some of your homework just because I have so many unexpected things that I have to do, but I will get that done and out to you. I am noticing that your work on the DNA purification, the very last lab we did, your work on that activity, particularly the calculations of the amount of DNA that was isolated, is so far everybody has that wrong. So we might have to go over that pretty soon together, and I'm thinking this Thursday might provide an opportunity. So keep it in the back of your mind that maybe we spend a little bit of time together on Thursday going over a couple things just for the good of everybody. Okay, I'm going to stop sharing my screen, and I'm going to ask you if you have any questions before we stop. Jose asks, do we have to know the different amino acids? No, you don't have to know them by memory, Jose. If I need you to know amino acids, I'll have it on the exam. If you ever take a formal biochemistry course, you will have to know the amino acids from memory, but not for me. Gang, do you have any other questions? Kevin, I sent an email to you before class. If you'd please check your email today.
Regarding the Mathematica notebook.
Yeah, exactly.
Okay.
Okay. And so, gang, I'll stay online, and I want to take a little break, but I'll be back about 2:10. If any of you need help, if you have questions about anything, I'll help you in about 15 minutes. Okay. So until then, if you're logging off, take care of yourselves. Be safe. It's getting a little crazy out there, and I will see you on Thursday. Be back in 15 minutes. Okay. Hayden, are you still there?
Yes.
Okay, what can I help you with?
Um, so on your illustration that you did last lecture when you had a top and bottom gene and you were describing transcription. I was just a little confused on your questions. Your first question, you said, which strand of DNA contains the gene? And you said the bottom strand, the template, contains the gene. But wouldn't, let me turn my video on. Wouldn't both strands contain the gene because they're partners, they're matches for each other?
Yeah. No, you might want to mute your mic there.
Okay.
Only one strand of the DNA helix contains the actual gene because it's the strand of the DNA helix that is the template for RNA polymerase to do its work is the one that gets decoded.
So what's the other side then?
Well, the other side honestly could have a different gene sequence for a different RNA and a different protein. So let me see if I can find that picture again, and I'll try to bring it up for us here. Hold on a sec. Okay. Okay, can you see the PowerPoint at this point?
Yeah, I can see it. Sorry, I was muted.
Okay. Okay, so yeah, did my answer make sense to you? Even though DNA has each DNA molecule has a left and right side, right? A top and bottom, it's a double helix. Only one strand is going to actually be decoded by the RNA polymerase. So whichever strand is actively being decoded by RNA polymerase, that's the strand that actually contains the gene information to make the messenger RNA.
But I know you said the other side could be a whole different gene, but it's literally its match.
Right, okay, so let me see here. Do you notice where I'm circling right now with my cursor? Here's the messenger RNA sequence. And you notice that the messenger RNA sequence is basically the same as the top strand of DNA. Okay, RNA uses U where DNA uses T. So U and T are basically the same. C and C is the same. A and A is the same. Then we have the two U's and T's. Then we have A and A and A. What I'm saying is that this messenger RNA could not have been manufactured by using the top strand as the sequence. It had to be manufactured using the bottom strand as the template to make this messenger RNA.
Yeah, I get that.
Okay.
Okay.
So you know, it's a matter of changing some of the preconceived information you might have in your head about what a gene really is. A gene is not literally both strands of the DNA. It's going to be one of the strands of the double helix. It's not going to be both strands of the helix.
Okay. Yeah. Okay. So I have clarity now. Then the second part, you asked, what's the chemical directionality of the top strand? And you said 3 to 5. Is it 3 to 5 because the bottom's 5 to 3? Like, aren't they anti-parallel?
Yes. Yes, that is exactly correct. Okay. So if the bottom strand is the template for RNA polymerase to manufacture the RNA, and if the directionality of the bottom strand is 5' on the left, then that means the RNA made from it has to be 5' on the right.
Why?
Well, because because RNA polymerase always makes RNA in the opposite direction of the template strand, 5' to 3'.
Oh, okay. Wait, I thought it makes it in the same direction if it's the leading strand and then the lagging strand goes in the opposite direction.
Well, okay, so now you're starting to confuse some topics, because when you start talking about leading and lagging strands, you're talking about DNA replication, and right now we're talking about transcription.
Okay. Can you say that again? Like describe the process of it replicating?
You want me to repeat what I just said?
Yeah, and why it's going in a different direction?
Well, so always keep in your mind, get that burned into your brain for your exam, that DNA and RNA polymerase enzymes, they always synthesize in the 5' to 3' direction. 5' to 3' means the direction of the molecule that's actively being synthesized, that's actively being made, which will be exactly anti-parallel to the template strand. So if the bottom sequence on your screen is 5' on the left, then that means the anti-parallel sequence has to be 3' on the left.
Okay. Yeah, I didn't know it was anti-parallel. I don't remember reading that. Because when it's making it in the video, it looked like it was going in the same direction as the
Okay.
DNA.
Let me see here. Let me see if I can find a picture in your textbook that might help you. Okay, if you can look at figure 12 on page 189. Figure 12, page 189.
I don't have my textbook right now with
Oh,
me.
shoot. Okay, that's going to make it hard. That's very hard, Hayden.
I can grab it in like less than 10 seconds though.
Okay, go ahead.
I'll be right back. Okay. What page is it?
Page 189, figure 12.
Yeah.
Okay. So just
Wait,
so
but
you know, for figure 12, we are talking about DNA replication right now. Okay, and the PowerPoint picture that we've been talking about is about transcription. Even though we're changing topics, the concept that I think you're confused about is still universally true for DNA replication and transcription. And the part of it that is universally true is that synthesis of DNA or RNA is always 5' to 3', always 5' to 3'. So if you look on page 189, Hayden, you notice that there's two pink strips of DNA. One's labeled the lagging strand and one's labeled the leading strand.
yeah.
Okay. Now tell me, what is the directionality on the left side of the picture for the lagging strand?
It's 5' to 3'.
Correct. On the left it's 5'. Now, Hayden, what is the directionality on the left side of the leading strand?
3' to 5'.
Okay, so that reinforces the idea that the two strands of DNA are anti-parallel. Does that make sense to you?
Yeah.
Okay. All right, now let's take a look at the leading strand of pink DNA at the bottom there. we've already established that it runs 3' on the left to 5' on the right. So if that leading strand is used as a template to make the blue piece of RNA right above it, what's the directionality of the blue fragment on the left side?
5 to 3.
5 to 3. Do you see that the fragment that was made in blue is 5 to 3?
Yeah.
Is that consistent with what I'm telling you, that synthesis always occurs 5' to 3'?
Yeah.
Okay. Now where the confusion comes in, if we're going to have the same conversation, let's go to the lagging strand on top. The lagging strand on top is already 5' on the left, which means that the DNA polymerase in this case needs to manufacture the blue fragment 5' to 3'. Okay, that first blue fragment at the top of the lagging strand, where's the 5' in relation to the blue fragment?
It's on the right.
It's on the right. So if the enzyme were to do its work, the enzyme would be moving in the opposite direction as the replication fork. Does that make sense?
Yeah.
Because that can't be allowed. We can't have the enzyme moving away from the replication fork. The only way to get the enzyme to move in the same direction as the replication fork and still be synthesizing 5' to 3' is to put a loop in the lagging strand of DNA.
Yeah.
So when you loop the lagging strand, you can get the enzyme that's making that new snippet of blue to move in the same direction as the bottom enzyme is moving and still be synthesizing 5' to 3. It's the looping of the DNA that allows the enzyme to move in the same direction as the replication fork and still be synthesizing that DNA 5' to 3'.
Is this fork bidirectional?
Well, there would be 2 replication forks on the circular bacterial chromosome, and figure 12 is only showing you one of them.
Oh,
So
okay.
there would be another replication fork moving off to the left that is not drawn.
Okay. Okay. I think I understand the picture we have in our notes now.
Okay. I would really encourage you to sit down sometime when you have some time and literally draw figure 12 out. Draw it out on your notes and color code it, and pay attention to the directionality and make sure you understand in your mind why the 3' and 5' are at the positions they are.
Okay. Another question. Why is the promoter code on the top strand and not the bottom strand?
Well, this could go back to you not getting emails from me. Are you definitely not getting emails from me?
I do get emails, but I didn't get your study guide. It goes from like the 24th to the 27th in my emails.
Okay, and it's definitely not in your spam folder?
No.
Okay. Please send me another email today and just ask me to retransmit some documents to you. With the same email that I sent out the study guide, I also sent out another Word document where I go over some of the questions that you're currently asking me. And so
Okay.
I think that document, if you can read it, might help you understand why things are the way they are.
Okay. Before you go, question 3, it starts on the RNA polymerase starts on the right side because our messenger RNA, it goes from 5 to 3, so it has to go from right to left.
Not really. when you look at the picture here that I'm circling, you notice it has this -10 region and it's -35 region. This is the region of the promoter, and the promoter is always the binding target for RNA polymerase. So that means since this region is on the right side, RNA polymerase had to sit down here on the right side.
Okay.
Wherever you find the promoter, that's
Oh,
where
I can't
RNA
hear you
polymerase is
anymore.
going to start. What else can I help you with?